Hello, my name is Paul Marcogliese and I'm a postdoctoral associate from Baylor College of Medicine. In the Yamamoto and Wangler labs, we employed genetic strategies in fruit flies to determine the functional consequences of de novo missense variants identified in autism spectrum disorder. While there has been many large-scale efforts to sequence ASD cohorts, there is a need in providing functional information for these variants. We filtered ASD variants from the Simon Simplex collection based on gene conservation and tool availability in flies. We looked for intronic mimic elements. In short, these are transposable elements that we can then swap out for any cassette we desire. We replaced these elements with an artificial exon called the splice acceptor T2A gal4 poly A tail, termed TG4. In these mutant flies, the endogenous fly gene gets truncated, acting as a strong loss of function. Importantly, a gal4 is generated in the same spatial and temporal pattern. This allows us to replace the fly gene with human cDNA expression and compare reference and variant directly. Thus, we also generated transgenic flies for corresponding human reference and ASD variants. We generated over 300 fly stocks and the ability to assess 79 SSC variants. We then characterized the fly TG4 mutants for either lethality or behavioral defects. In parallel, we crossed these lines to a UAS-GFP reporter to determine the CNS expression of these fly genes. We then humanized our flies in a rescue-based strategy to determine if reference or SSC variant could rescue lethality or behavior defects that we found. We also used a tissue-specific overexpression-based strategy to complement our rescue-based strategy. The detailed outcomes of each strategy are contained within the poster, but in total we found 30 SSC variants of 79 tested that showed a functional difference from their corresponding reference. Given the overlap of ASD genes with other neurodevelopmental disorders, we looked for other patients for some of our gene hits. With our collaborators, we identified patients with a neurodevelopmental disorder with variable presentation with variants in the X-linked gene GLRA2. Intriguingly, we found a recurring de novo missense variant in 4 female patients at threonine 296. Further studies in flies showed that de novos in female patients acted as gain of function, producing morphological defects and increased synaptic transmission, as opposed to maternally inherited variants found in males that acted as loss of functions. Our study combines sophisticated fly genetics with clinical collaboration to identify a new human disease gene. Please see the poster or preprint on bioRxiv. Thank you.